I'd like to introduce next Benjamin Wylie. It's my pleasure today to introduce Ben Wylie. He has an all-star training, first in the lab of Chad Rienstra at University of Illinois, Urbana-Champaign, doing things like tensor correlation, CSA, and so on. And then he did a postdoc with Anne McDermott, where he studied membrane protein ion channels. And he has interests now in membrane proteins, including GPCRs, and still continuing with ion channels, which is what he's going to tell us about today on a different lipid-regulated ion channel. And he's now a professor in Lubbock, Texas, at Texas Tech University. So with that, this virtual floor is yours.
Thank you. There we go. So thank you for having me. So yes, I do have interest in GPCRs, but that story is actually going to be coming the next year. What I'm going to talk about today is actually continuing on with ion channels, specifically different lipid regulation mechanisms of the lipid ion channel KirBact 1.1, or the lipid regulated ion channel KirBact 1.1. This is a bacterial homologue of eukaryotic channels, so it's going to be similar to a lot of these ion channels that are gated by PIP2, and I'll show you pictures of that in a minute. But just like all potassium channels, it's a homotetrameric protein, and it then is going to be able to transport through diffusion potassium across the membrane. It's inward rectifier because it actually tends to pass potassium in as a preference for that rather than out. And it does it through the membrane based upon two coupled gates, right? And one of the things that we're going to tell you is that these actually are coupled, even though these channels are constitutively open in vivo, okay? And so the first gate is just the selectivity filter, just these special, and this is of course very similar to the selectivity filter in KCSA, which I believe Anne McDermott told you all about a little over a month ago. And so you basically have this unusual arrangement where you have carbonyls pointing in the same direction that are then creating a similar coordination environment that potassium would absorb in water, thus allowing the potassium to move through the selectivity filter based on either a hard or soft knock-on mechanism, depending upon which side of the egg you want to start peeling from. I try to avoid this, but the difference being whether or not water is actually involved in that. And then all of these channels are going to have some sort of hydrophobic block at the intracellular interface, it's the plasmic interface, and they're going to respond to some stimulus, right? So of course there's a lot of discussion about, say, voltage gated channels. These channels tend to be ligand gated, specifically something anionic. It can be a lipid head group, or it could be something like ATP. In this case, it's going to be an anionic lipid head group. All right. So the reason I chose KirBact 1.1 is that it was bacterial, but it had a really high homology to eukaryotic channels. And you can see they have nearly identical folds. The big difference being that you have this large cationic patch that is directly below the inner leaflet of the membrane. And in the case of KirBact 1.1, it's smaller, right? And it's smaller because it's going to accommodate a smaller lipid, right? You do not have these, large inositol lipids in bacteria, right? So you have a similar gating mechanism, as I'm going to tell you, but it's going to be triggered by a smaller head group, right? But we do have actually eukaryotic versions of this channel of these channels expressing out in the lab too. So in the future, in the coming years, we're going to be telling you more about that. So obviously, I'm a solid state NMR guy. But I do try to pay attention a lot to the biology, and so we're doing everything here in liposomes. So this is all going to be reconstituted in the liposomes, and so we want to make sure that we're doing all of our NMR in a functionally relevant environment, right? So we've adapted these two different assays as a means of confirming that we are in fact in a active conformation of our channel in order to do our NMR, right? So we know what state we're in. And so the first assay we're doing, this was actually brought into the potassium channel literature by the group of Rod McKinnon, who of course won the Nobel Prize for first elucidating the structure of the selectivity filter of the potassium channel. And the idea here is that we're going to reconstitute our channel into a liposome with an identical ionic concentration of potassium and sodium on the outside. So there's no net voltage across this membrane, but there's also no ionic passage either because a potassium channel cannot conduct sodium. Then we are going to dope our liposome with this ACMA dye, which is sensitive to pH, right? And so then we take an ionophore that's going to bring in H+ cations. So then that's going to basically create that voltage gradient, right? So it's going to bring in the H+ cations, it's going to push out the potassium cations, and in the process, it's going to quench our dye, right? So that allows us to actually look out and get real-time real-time potassium flux without actually having to learn patch clamp, right? So we had this, this big ambition for a long time to actually like learn electrophysiology. This stopped me with my delusions of grandeur, right? So we're able to see this, and so we have a maximally gated environment here, okay? And this is actually important that it doesn't go to like full potassium flux, as I'll tell you in a minute. But then if you actually add the native ligand for eukaryotic channel, it starts to shut that flux off, and that's because it actually binds to the same site, but because that cation patch is smaller, it's actually going to lock the channel in place. It can't actually pull it open, right? And so then we also are doing fret, and so we have these fret labels that are at the tips of these C-terminal domains, these paddles, as we like to call them, right? And so we can actually then observe gating motions, and so we know then when our channel gates. And so we use these all the time when we move to a different lipid environment to confirm that we actually are doing this in something that is actually functional, that's something that actually changes its conformation consistent with a gating process. All right, so questions on the last talk about C13 assignment strategies. Well, that's all I do. And so, this potassium channel is pretty large based on solid state NMR standards. Each monomer is roughly the same size as the GPCR that Stan Appel did all that beautiful work on a few years ago. And so, proteins of that size getting complete assignments is difficult, especially since I just have a 600. And so we adapted non-uniform sampling, right? So of course there's many, many contributions to this field going way, way back, but we're actually adopted the treatment that Dave Robniak evangelized me at ENC a few years ago, using this double exponential weighting. So we're basically gonna do non-uniform sampling, but we're doing this double exponential bias that is going to dramatically improve our signal to noise, okay? And so I should also give a shout out that a lot of this was done using the NUS tool NMRBox. NMRBox has been a fantastic resource for us, and I'll show you some more stuff that they've helped us with in a couple minutes. And the data here, this is actually of the eukaryotic variant of the channel I'm telling you about today. Work that I'm hoping to be able to share with all of you in the coming year, but just work on the 800 at MAGLAB. In 2 hours, you can see that we're getting a factor of about 2.5 increase in signal noise for an NCA2D compared to uniform sampling. So to give you an idea of the power here. So applying this on our 600 back here in Lubbock, we're acquiring the three basic, I would say probably the three most basic backbone and sign chain assignment experiments in solids, which are the NCACX, NCOCX, NCA, and COCA, where we're going to skip this CO, and so we're going to have this combined direct detection emission that's going to include the CO and the CA. All right, and so I think everyone knows what backbone walks look like. And so I'm just showing you here to show the fact we actually did do backbone walks. But the interesting thing about this slide is that we started to get enough sensitivity and resolution to find that not only could we actually see all the residues, which was a huge plus moving to non-uniform sampling, but we actually found that we had two different conformers within that transmembrane region when we're in this activating lipid environment, this rich in POPG, right? And so this is just an example of, we could call this chemical shift perturbations, but these are chemical shift perturbations that are within the same sample, within the same environment. And we have them mapped out here. Okay, so we have this, these maps basically showing that when we open the activation gate here, and I don't have that structure to show you today, but it's going to be forthcoming. And so you basically at the opening of the activation gate here, you are going to find, wait, are you guys able to see my arrow?
Yeah.
Yeah, okay. Okay, good. So if the activation gate opens, then you're going to see kinks along the inner transmembrane helix, which is expected. This is something that's been observed in a lot of channels, obviously. But then we actually start to see perturbations near the selectivity filter and in the pore helix that's packed behind it. Okay. If we go on and then look at this in a purely zwitteronic environment where you know it's closed, here we find only one conformation. It's a different conformation from the two conformations that we observe in a gated environment. But then we start to see, if we compare that to the dominant conformer in our gated environment, we start to see that we have a huge number of, based on these perturbation maps, we see a huge number of conformational changes going up from the gate, the inner gate, all the way up to the turrets and the selectivity filter loop and all those places in the protein. All right, and so this was a little bit unexpected because unlike things like KCSA and like voltage gated channels, these channels are not known to undergo C-type inactivation. They don't spontaneously shut off when activated. And so we're trying to understand what that would mean. But by carefully examining our chemical shifts, we were able to come up with this model in the gating environment whereby it does appear that this fourth binding site, so it's understood that based on the knock-on mechanism, you need these four sites to be intact to have really efficient potassium reductance. That in this minorly populated state, that fourth binding site actually comes and goes. Okay, and it turns out based upon our NMR data, it does appear to come and go on roughly the same time scale as this phenomenon that you observe in the Kerr literature called fast gating, right? So what we might be observing here is sort of a secondary mechanism, is that these channels will be constitutively open under the right conditions, but there's actually means where they can actually sort of, adjust the spigot per se to like actually regulate the flow as needed, okay? But then if you compare our most dominant conductive conformer to the closed state, we start to notice that we have, this really interesting network of hydrophilic residues that are positioned between the first transmembrane helix and the second transmembrane helix and the pore helix. And they appear to all rearrange a hydrogen bonding network as a function of these gating phenomena, right? So we're actively creating mutants and trying to understand how this is all taking place. And this is really interesting because around the same time, almost exactly the same time we published this, the group of Bert de Groot came out with a similar study and using molecular dynamics, finding that some of these same features were preserved in the MTHK channel. So it turns out that we are probably observing similar regulatory features that are pervasive in potassium channels that don't undergo C-type inactivation. All right. Now we're interested, of course, in looking at the full channel as it's gated. And of course, I just talked about the chemical shift assignments in the transmembrane region. It turns out now we have chemical shift assignments from residue 1 all the way to residue 300, right? So we don't quite have the last 30 residues, but we've got most of the protein. And so what we found is that when we did all the assignments of the C-terminal region is that they didn't change. There was not a big conformational change. So we know from the fret that it's moving, right? So we wanted then to understand the motions. And so the first thing we did is we wanted to confirm the activation gate. And so through a lot of trial and error, we were able to identify these three arginines that appeared to be, and by trial and error I mean with like lots and lots of mutagenesis, lots of assays, these three arginines that appeared to be the ones that are primarily at play in this whole lipid gated mechanism, right? And they happen to be in a similar place to the a lot of the residues that are actually known to bind the PIP2 ring in closed but PIP2 bound structure of KIR 2.2, which is in this case, a channel from chickens. So moving on from there, we created this triple Q mutant. So we mutated those arginines to glutamine to try to minimize structural perturbation while like eliminating charge. And we can find here that in the presence of anionic lipids, that so-called triple Q mutant does not contain these arginine outliers. And these are actually the first like major structural clues I found when I first started my program at Texas Tech, is that I had these weird arginines that I didn't expect. And this is actually one of the first data sets that I ever acquired at Tech, like many years ago. And you can see then in zwitteronic bilayers, for the wild type, we see almost exactly the same signal that we do for the mutant. So it does appear that these are the arginines that are, in fact, mutating those arginines away. It does appear that those are the ones that are binding phosphates. We wanted to further confirm that those were phosphate contacts. So we went to REDOR, right? We're doing carbon phosphorus REDOR. And so, in fact, those arginines dephase, right? Those C-zetas dephase. And of course, there's three of them, and we weren't sure if there's one phosphate or two phosphates, but it was still pretty clear based upon the simulations we did that all three of those arginines would be within a distance consistent with some sort of salt bridge to a phosphate. So we know that then, okay, we've confirmed that this is in fact the locus of anionic lipid binding. Therefore, it's like, say, if you want to think of this like the active site, but we want to understand what the gating motions are going to be. And we know that the chemical shifts aren't going to help us except for maybe as markers, but they're not going to give us like structural clues. So of course we went to water-edited spectroscopy. And for those of you familiar with this literature going way back to the 70s, and you've read, of course, Klaus Schmidt-Rohr's book he wrote with Hans Spies, these, experiments reach for domain motions. But of course, we were inspired by this by the work of Mark Baldus. And of course, then May Hong has done lots of beautiful work using water-edited spectroscopy. But the idea here is we're using this for domain motions. And so we're going to do we're going to excite on proton, do a T2 filter, okay? And the T2 filter, we're in solids, right? Our proton T1 or T2s are really, really short, right? So they're going to go, so all the protons or the protein signals will go away. And then we're going to have spin diffusion from the water bath to the protein. And so then only things that are water proximal are going to survive, okay? So like I said, the chemical shifts are markers. And, I assumed naively going into this that we were going to see what Mark Baldus saw, right? That the water accessibility increases when the channel opens, and it turns out that was not the case, okay? When the channel opens, of course, things that are near that activation gate become here in blue. So in blue is the gated wild type, okay? So all the things that you would expect that have to become water accessible when the channel gates do in fact become water accessible here in blue. But these things in red that are in the C-terminus, actually the C-terminus is far more water accessible, especially charged residues, when the channel is not gated versus when it's gated. So that was sort of contrary to what I was expecting. And so we actually quantified this, and this, Lauren Andreas, of course, published a really beautiful paper looking at this under faster spinning. We took their formalism, basically added a protein-protein diffusion term, fit that, and we found that then these ionizable side chains, especially arginines and glutamates, which we had nice signals on because they're outliers, are far more solvent accessible in our closed dead triple Q mutant here in red versus the wild type here in black, okay? And the solvent accessibility surface actually increases when the channel's closed compared to when it's open, all right? So from that, we were able to figure out that the open state of this channel actually is probably stabilized by a bunch of these ionizable side chains that are going to be buried, right? So basically what happens is when the channel gates, these paddles are going to rotate, and they're going to basically form these salt bridges between subunits, and those subunit salt bridges then help to stabilize the activated state. And that makes sense, right? If you think about, a PG lipid or even cardiolipin, something like that binding, that's actually not going to produce a whole lot of force. Something that it's going to have a helper, and the helper is the formation of all these interdomain salt bridges, right? And of course, we are actively working on solving a structural model of this. We've got the samples made, trying to find those salt bridges in order to pull the channel open, all right? We are working on full-length models of KirBact 1.1. So this is actually our full-length version of the closed state. You can see here that this is actually the crystal structure. Lots of domains regions are missing. Using a combination of chemical shifts and other means, we're actually are closing out on a nice model. And of course, Charles Schwieters has been really helpful in helping us do a lot of this. So the last story I want to tell you is sort of what we've been doing in shutdown. We were obviously shut down for a while during quarantine. And so we actually had a study that we published in BioPhysJ last year where we talked about how KirBact seemed to actually have these really profound properties on the membrane to which it was reconstituted. And we postulated it's because that these CARC motifs were conserved, and these actually exist in a lot of eukaryotic KIR channels. Okay, and probably they're there because they're native hopanoids, these polycyclic 5-member green lipids that exist in the native organism. And so if you actually then perform these assays I told you about, but now we're actually going to have it in a maximally POPG gating environment. Right here, we start adding cholesterol. The cholesterol actually starts to increase the activity at low concentrations, but then as the concentration goes up, we start to lose potassium current. This is known that cholesterol can actually be an activator of a lot of eukaryotic channels. And so then doing our FRET, you can see that cholesterol does not seem to at all change the conformation of the C-terminus when it's added to a POPC environment. And then as you add cholesterol to that gating mixture, you actually then start to converge towards the closed state, all right? So moving on from here, this is at this point we were at this stage. Then we have to go into quarantine, right? So we became the Wylie Computational Lab, and so we started doing a lot of Martini simulations, and we found then in a cholesterol-rich bilayer by Martini that these areas that we identified by homology seem to actually be the regions that in fact bind cholesterol, right? And they happen to be sort of located near where you would have the anionic lipids would need to be able to access those arginines, okay? So moving on from there, we also did some atomistic simulations, and these aren't completely finished, but we actually started to notice that in our atomistic simulations, we found cholesterol dimers and even trimers that start to form on the periphery of the protein, right? So this was something we sort of predicted, right? We were joking that we didn't have a lipid raft, we had a lipid life vest in certain lipid mixtures around our protein, and the MD seemed to bear that out. And into this point, then we had, what we had done is we'd made all these glycerol labeled samples of KirBact that we had reconstituted with also skip labeled samples of cholesterol. In this case, we're using my graduate advisor, his grad student, Del Ripa. They came up with this 2-acetate skip labeled version of cholesterol, and putting those together. At this point, I'm shut down. And so what's great is not only having a great graduate advisor, but having a great former graduate advisor. As you know, Chad has moved to NMR FAM, and so that we were given priority access at NMR FAM for solids. And so here are spectra. This is just skip labeled cholesterol at 250 milliseconds of mixing in just lipids. And then we have the skip labeled cholesterol with our protein here in blue. And you can see that we have splittings, right? So we knew that we had 2 different populations. But it turns out those 2 different populations, not only were they conformationally distinct, they actually were in contact with one another. So the dimers that we were observing by MD happens to be also present in these samples, okay? And so we actually, I think we all know how the structures, but we have we've worked out the distances. We're working on the conformations here and everything. But we can actually find, as I said, in the MD, we've actually found these dimers that seem to be creeping into that anionic lipid binding pocket. And we've even found some of these trimers that are sticking to the outside of the protein. But then based upon all of our NMR measurements that we've been performing in NMR FAM in collaboration with Chad and his graduate student, Eric Hahn, we actually have been able to, this is now this is preliminary, we're finalizing this, and hopefully we'll have it submitted soon, but we actually are able to find that this cholesterol dimer forms, and it actually blocks pretty readily. I mean, we're able to see, very strong spectra of this, but it seems to dock in that pocket. And so this sort of helps to explain and perhaps why you have to reach a certain cholesterol concentration for it to become an activating, is because it has to reach a concentration where those dimers can actually form. And so this is something that we're really excited about and will be submitted in the coming months. And so, sorry I went 2 minutes over, but with that, I'd like to thank my group, especially my, as you can tell from the citations, my 2 most senior grad students, Colin Borsig and Razamani, that this was a team effort, and Derek and Mariam and Isaac especially played important roles. I really want to thank Chad and Eric for giving us access to NMR FAM and all their help. I also want to thank MAGLAB for helping us with a lot of related work. And of course, NMR Box, where we've been able to run lots and lots of simulations. So with that, thank you for your attention. I should also thank the NIH. Thank you for letting me pay for this.
Thanks for the fantastic presentation. As always, please place your questions in the Q&A when you have them. Since there aren't any there yet, I'll start. When you have the liposome samples for these proteins, I'm just curious, with some other membrane proteins, you see interesting features in the shapes. Have you looked at the just negative stain EM?
Yeah. Oh, no, we have negative stain. Actually, so my students laugh at me because I put things in I leave things in overflow. And oh crap, I actually did cut those. I have, if you are at all curious, Lauren, I have in that BioPhys J paper, I have all the negative stain EM images of liposomes. And so all of our samples are, they all start off as what would be on the small end of large unilamellar vesicles. They're all around 200-ish nanometers. We do typically though, and this was actually something that we started doing when I was at Columbia. We do often find that just magical spinning applies enough force that you'll drive into multilamellar vesicles anyway. So we often will do that. And so we actually then we didn't publish those images, but there are images like we'll form a big union all of us. We drive it to that condition anyway, and then that actually also gives us, you know, higher signal to noise, higher protein packing, but also better order. So, but yeah, we do that. We absolutely do the EM.
Thanks. And so far, I have just thank you for sharing interesting data from Christoph Grohe. Then, since there aren't any other questions yet, well, oh, there is one now. Would it be of any advantage to use proton detection, for example, for proton accessibility? From Christoph Grohe again.
Oh, yeah, we're actually doing that right now. Yeah, so we actually have, yeah, so we have deuterated and back exchanged KirBact, and we are running that right now on the 900 at NMR fam. We actually started doing that on our 600. Even though our proton lines are okay, it was still kind of a big oval, so the 900 has been really helpful for that.
So is that with a deuterated sample then?
Yeah. So we have it, so it's deuterated and then about 25% back exchanged. And so we're actually doing that as we speak.
Are you able to back exchange most of the residues of the protein? I know a lot of people have concerns about membrane proteins with back exchange.
So the sample that we have is in the it's in POPC, so it's going to be the closed state. And of course, then we've seen almost everything in the C terminus, and then we see a lot of things near the turrets, right? And actually, you do get, you know, there's a lot of trapped water in the middle of the channel, where we really don't see anything as along transmembrane 1. So that transmembrane helix 1, you see actually no back exchange, but you do actually see a fair amount in the C terminus, and like the very central part of the cavity, and then of course near the top.
And I have a curiosity. Normally, what I've seen in potassium channels for measuring the activity is patch clamp experiments, but you show different experiments. Is this because of where you embed the protein and you cannot do patch clamping there, or just?
Oh, no, we could do patch clamp. It's just I don't know how to do it.
Oh, okay. Because
I have been meaning to do this for like basically any day now. I'm going to go across to the med school and patch clamp this, but honestly, because we we started off doing rubidium uptake, and that was a big pain in the butt. So we once we started using this assay, because we could actually get rates in real time, it was satisfactory for our needs, right? Sure, but we do have an interest in doing that. We just haven't done it. But there's no reason we can't do it. I mean, we're using very similar. Like of course, like Colin Nichols does lots of patch clamp of this channel, and of course we're using very similar conditions to his, so we could easily patch it. We just haven't.
Okay, thank you.
Marcus had a question.
Yeah, so I was wondering about your cholesterol binding. I mean, how specific is that in, let's say, in the simulations as well as in the chemical shift perturbations? I mean, is this really one site, or you have other sites, and is there a correspondence between the Martini simulations and the NMR, or the Martini just gives you a whole zoo and so on?
Well, so I mean, so the Martini simulations are not exactly the same as the NMR, right? But they gives you the same ballpark, right? So we do have, so of course, as you might imagine, a lot of these, because we're looking at, this is, we have a lot of methyl groups, we have a lot of aromatic side chains, therefore there is a lot of ambiguity, right? And we're actually actively using tools within Haddock to sort out the ambiguous restraints. And of course, eventually we'll run production MD with those restraints. Or actually, eventually, I mean, actually very soon. But, so you would see there's actually a fairly high correspondence between that and where we're finding what Martini would predict. Okay, so basically here, we are seeing a pretty high affinity. Okay. Now there does seem to be a less tightly bound but still existent cholesterol binding site up here by NMR. Okay, but those distances are far weaker, right? And that's the thing about this dimer. This dimer is really tightly in there, and so we're actually seeing like not only are we seeing really tight distances between the two cholesterols, we're seeing really tight distances between that dimer and sites on the protein. Whereas the one that's up here is actually like far weaker. So it's, I wouldn't say it's non-specific, but it's not as strong.
Okay, thank you.
And on the same slide, I'll ask another thing,
Okay.
because is this, the KirBact 1.1, if I got it right, is bacterial, right?
Mhm.
This is the back. So, I mean, most bacteria don't have much cholesterol. Some get it from host systems, but then what is the role of cholesterol for the bacterial homologue here?
Well,
it's sterile?
so cholesterol. So okay, so the cholesterol in our bodies. So in the channel, in the cousins of this channel that exist in our bodies, the cholesterol is known to be a secondary regulator, right? And that cholesterol is capable of actually turning off potassium current in these channels. Now, of course, it's far more complicated because you have sphingolipids, you have all these other things, right? So in vitro, it's very easy to get the shut off time, whereas in vivo, it's a little bit more complex. But KirBact, the reason I picked it is it's so homologous to a eukaryotic channel that actually almost exactly the same binding pocket. And this has been identified by Levitan and by Colin Nichols, and actually Samson has done a lot of MD on these channels recently. But that cholesterol binding pocket actually is interestingly conserved, and that's probably because there are hopanoids in Thailandensis, which is the native organ, the Thailandensis species, which are native organisms for these channels, have hopanoids, right? And so they actually have polycyclic lipids that are similar to cholesterol. So we are probably, so we're basically in that way, we're kind of lucky in that cholesterol and hopanoids are probably fulfilling similar roles in these organisms. And so we're actually then able to look at something that we know to be a feature of eukaryotic biology, but using our prokaryotic system.
I see. Is that, are those lipids also an option for studying in the future?
Oh, actually, yeah, no, we have those. We have those expressing. In fact, we just got a grant from, or a supplement to our grant from NIH to get an HPLC to purify all of those. But yeah, we have actually 2 different hopanoids that we have expressing in a more quantity. HPLC will be installed. We had access to one, but then the guy retired. So then our own HPLC will be installed actually the week of Thanksgiving because, timing, 2020 and all that. But once we have that, then actually we'll have pure hopanoids, and we can then do all those comparisons, right? And the interesting thing about the hopanoids is that these are all well, with exception, we use the Burkholderia thailandensis as a model organism, but a lot of the other Burkholderia are pathogens, and actually these hopanoids, there's a secondary interest too, in that the hopanoids are thought to confer a form of antibiotic resistance. They help to keep those membranes a little bit more impermeable to certain classes of antibiotics. And so there's a lot of health relevance for human health there too, actually. So.
Okay, thanks a lot. Well, I don't see more questions in the Q&A. So unless more panelists would like to ask something, then let's go to the informal session. Thanks again.
Thank you.
Both speakers.